Good afternoon. My name is Dr. Maricica Pacurari. I'm an associate professor in the Department of Biology at Jackson State University. And today I'm here to talk to you about protein glucosamine in acute lung injury. Acute lung injury is an acute and persistent lung inflammation response caused by direct or indirect risk factors such as sepsis, inhalation injury, trauma, massive blood transfusion, and pneumonia. A hallmark of ALI is excessive inflammation of alveolar interstitial nerve space, infiltration of neutrophils, and secretion of the cytokines, chemokines, and proteases. Clinically, ALI is manifested with severe tachypnea, declining oxygen pressure, hypoxemia, and pulmonary edema. Lipopolysaccharide is a gram-negative membrane component and a model to study ALI or acute respiratory distress syndrome. Is there a need to study ALI or ARSDS? Yes, of course. The incidence of these two is about 190,000 cases per year with 70,000 deaths per year. Therefore, understanding the molecular mechanism of injury-mediated signaling may aid in the design of novel therapies that may alleviate the exacerbation of lung inflammatory responses. What is MP? MP, macrophage inflammatory protein 4, or the same time chemokine ligand 18, CCL18 was discovered in the 1990s. It is highly expressed in chronic inflammatory lung disease, and it is produced by alternatively activated macrophages. Glucosamine is an amino sugar building block for the glycosaminoglycans, abbreviated GAGs. GAGs are linear long polysaccharide of repeating disaccharides that can be either acetylated or sulfated, represented by hyaluronic acid, heparan, chondroitin sulfate, keratan sulfate, dermatan sulfate. In the lungs, GAGs have an array of functions such as support structure for the interstitium, subepithelial tissue, bronchial wall, regulate water homeostasis, modulate inflammation, and tissue repair and remodeling. Protein GlcNAcylation is a post-translational modification carried on by two enzymes, OGT and OGA, which add or remove the glucosaminoglycan on the serine or threonine residues. Glycosaminoglycation is an essential modification of proteins which plays an important with numerous functions, cellular functions such as cell cycle, transcription, signal metabolism, or proteases. The hypothesis for this study was that glucosamine attenuates LPS-induced increase in MP4. And here we have the results. So LPS at 10 micrograms significantly increased MP4, whereas treatment with glucosamine will reduce the increase. Immunodetection study indicates that we tested two doses, 10 and 20 of LPS, and we find that the difference between control and glucosamine was insignificant, while the LPS 10 and 20 increased immunodetection, while the glucosamine with both those two doses attenuated the immunodetection of MP4. As a mechanism to understand how glucosamine may attenuate LPS-induced activation of MIP4, we looked at NF-kappa B, and we find that in absence of glucosamine, there is an increase in immunodetection of B65. Another mechanism that we investigated was to look at whether the glucosamine increases protein GlcNAcylation in the presence of LPS, and we find that in the presence of glucosamine, the LPS increased protein GlcNAcylation. The conclusion of the study is that LPS is a powerful inducer of MIP4, and glucosamine attenuates LPS effects through inhibiting NF-kappa activation and increasing protein GlcNAcylation. This preliminary data suggests that glucosamine may contribute to reduce LPS-mediated effect on MAP4. I would like to acknowledge the support from NIH and MIDA 254 support for my research project, Johnson State University, my College of Science, Engineering, Technology, and the undergraduate and graduate and a research associate in my lab. If there are any questions, I'm happy to ask. Thank you for your time.